Um, so thanks everybody for coming, both committee members. Thank you for being here, the colleagues, family for coming out. It's been really my pleasure to have Shelly in the lab. She started um, as a staff member in my lab. She came from a collaborators group at Dana-Farber, and immediately she had a plan. She hit the ground running and said, I want to apply to UCSD. And I look, because I'm always trying to encourage people like her predecessor, Mary. Mary, you need to be in graduate school. She was like right on the ball. Before I even knew it, she had submitted an NSF fellowship, pretty much without my knowledge. Like, hey, Joe, I need a letter of recommendation. Like, for what? Lana writes it, gets it, right? And so, and then she's like, plows through her thesis work, publishes the major work in Nature Methods, gets the cover, okay, and then has a new plan. Okay, hey, I can do this thing, and I was like, okay, whatever you want. At this point, she's on a trajectory that I think would be hard for anybody else to keep up with, and I understand she's got a plan for the next phase, which maybe her committee will ask her about. So, without further ado, Shelly.
So, hi everyone. I'm happy to share with you today a couple stories about network biology and how omics tools allow us to get a high resolution perspective on what's happening inside of animals and cells as they develop or deal with environmental stress. I first want to take a second to dedicate this talk to my family and to my brother Scott. He was my main inspiration for doing my PhD research or trying to get my PhD, and I could not have done this without the torch he passed on to me, or also without the unconditional support of my family and my husband, Austin. All right, so during this talk, I'm going to cover some network biology basics. I'm going to talk about why it's important and some technological shortcomings that led to my PhD research. And then I'm going to show you how I developed a technology in Joe's lab to do high throughput protein interactome mapping. And I'll show you how I use that technology to unravel complex transcription factor regulatory networks in plants. And then finally, I'll tell you about how I used omics to assess climate change impact on a non-model organism. Okay, so when I say systems biology or network biology, I'm talking about trying to understand how a collection of molecules work together to make biological processes happen. And this is similar to how different car parts work together to make a car run. Cells are like cars in that they are complex systems with many working parts. And if we just know what the parts are, but not how they are connected, it makes it really hard to diagnose problems when they arise. Kind of like how fixing one part of a car doesn't always fix the whole problem, or how one gene is not always the cause of a disease. So how do we get this kind of network bird's eye view of a cell? We can use omics technologies. So omics technologies allow us to see a snapshot of all the biological components in cells, like how genomics allow us to see all the genes or genetic material in the cell. And these biological components work together to make biological processes happen, just like how all these components work together when preparing a meal. So the genome is like the big recipe for the cell. And the transcriptome, which is the collection of RNA, are like the orders that are yelled across the kitchen to recruit different sous chefs to come and cook. And each sous chef has his own unique skill, just like proteins in the cell. They all have these different skills, and they work together as a proteome, or collection of sous chefs. And then the sous chefs work together to kind of do these different tasks. So like grilling food or making a soup. And that's kind of like how proteins interact together as an interactome. the collection of all protein interactions. And then the outcome is this big feast, which is all these different types of food. And that's like the metabolome and all the different small molecules that are in the cell that result from all the protein interactions. I'm going to focus today on interactomes and metabolomes. But first I want to show you a couple of examples that show why I'm sorry, I'm excited. A couple of examples that demonstrate why the systems biology perspective can tell us a lot about biology. Okay, so in this example, done by Mukhtar et al. In collaboration with the Ecker Lab, they did this protein-protein interaction screen using a high throughput method and identified that these 18 Arabidopsis proteins were commonly targeted by proteins from these two different plant pathogens. And they then used this information to do a genetic experiment and look at how mutant plants responded to pathogen infection. So in the experiment, they found that the plant showed increased pathogen infection when 9 of these 18 proteins were mutated. So they tested 9 different plants, each having a different mutation. And 9 of them showed this increased pathogen infection. And then 6 of them, different from the 9, showed increased resistance to the pathogen. So overall, they found that from this interaction screen, these proteins were actually involved in plant immunity, which wasn't known before. And so you can see how using protein interaction data like this can be pretty powerful in designing experiments for crop optimization. In this example, this is a research study done in the Ecker Lab. They integrated protein interaction data with patient genomic tumor data. And in integrating the two data types, they were able to find similarly affected protein networks among different patients. And when they classified the patients by these different subtypes, they found that the different subtypes could actually predict how the patients responded to different treatment or how the patients would survive. So you can see how this kind of data integration can be powerful in things like personalized medicine. But there's still some shortcomings that exist in omics studies. And one is that the interactomics field is lagging. Time, cost, and sensitivity constraints have limited assay throughput for years in the number of protein interactions that can be screened at once and the number of replicate assays that can be performed. And these are some of the highest capacity assays that exist today. And you can see that they're able to screen thousands of proteins and identify a lot of protein interaction data. Yet the size of the proteome is so large that none of these technologies have actually been able to screen the number of interactions to get this proteome-wide interaction data that would be really powerful for the data integration. So you can imagine this combinatorial, so if we need to screen over 1 billion different protein combinations, we need to have a technology that can have exponential throughput and be easily replicated. And another shortcoming is that there's limited omics data for non-model organisms. And this is kind of a problem because omics data could actually help us assess things like climate change impact on organisms, which could then help better inform policies. And we're trying to, it would be great to try to understand which animals, are going to be around in the future to try to understand how we should be regulating things. Okay, so yeah, all those things led to my PhD research questions, which are, can protein interaction technology throughput be improved? Can improved interaction technology address the complexity of plant regulatory networks? And can omics data help assess climate change impact on non-model organisms? Okay, so I'm first going to explain about the yeast-2 hybrid system a little more. So this is a technology that I showed in the last slide, and it's the most economical technology out of the others. And it works by expressing different proteins in yeast cells. So say you want to know if the blue and the green protein interact. What you do is put the coding sequence of the protein into one yeast strain, and you put it in this plasmid that carries this activation domain. It's a yeast activation domain that gets put onto the protein. In another yeast strain, you express the green protein, which gets expressed with a yeast DNA binding domain. And then you mate these two yeast strains together to get the proteins in the same cell. Then, if the proteins interact, it brings the yeast activation domain and DNA binding domain together to form a functional transcription factor that's able to express this reporter gene. In this case, it's a reporter gene that allows the yeast to grow on this selection media. This way, you can select for cells that have protein interactions in them. If there's no protein interaction, the yeast cannot turn on this reporter gene expression and the cells don't grow. Okay. So one thing about this system is that, so if you can imagine you want to test many combinations, not just blue and green, but all different combinations, in order to keep track of which proteins are interacting, you have to spot each yeast cell with two proteins in it on separate places on a plate like this, and that way you know you can tell who interacted with who. So, and then you would visually judge which cells had interacting proteins in them based on their growth on the plate. But you can imagine that can be really time-consuming and labor-intensive to have all these plates testing all these different interactions. But if you combine yeast-2 hybrid with next-gen sequencing for interaction detection, you could really increase your ability to detect more interactions at once. So remember, if we are trying to test over 1 billion different protein combinations, in one NovaSeq run, we could get at least 10x coverage per pair if we use next-gen sequencing as a detection method. In reality, it's estimated that about 10 proteins in 10,000 pairs interact. So you could really get about 8,000x coverage. And there's a couple or a few different technologies that have been developed that do use yeast-2 hybrid with next-gen sequencing. In this approach, one protein is screened against a pool of proteins. And because the identity of one protein is known, you only have to use sequencing to identify the interaction partners from the pool. In this approach, you can do pool versus pool screening by integrating barcode sequences on the expression plasmids and then perform a recombination reaction to get the two barcodes on the same piece of DNA inside of each cell. And then you can just sequence the barcodes to find out what interaction was. However, these methods are still limited in their throughput. So the one versus pool method still requires thousands of screens to detect all the interactions. And that's not including the replicate screens that need to be done to make sure your data is reproducible. And the barcode technology still requires all these extra sequencing steps that become costly as you scale up. So Joe and I and Mary and others in the lab had this idea to add a Cree recombinase reporter gene into the yeast. And so what that would do is if there was a protein interaction, Cree recombinase would get expressed in the yeast cell, and it would recognize these mutant lock sequences on the expression plasmids and irreversibly recombine them to get the coding sequences of each protein on the same piece of DNA. In this way, this DNA sequence could be amplified, and you could use it to identify, or you sequence it and use it to identify what the protein interaction was. oops, sorry. Okay, so now this method, Cry2H-seq, which is Cree reporter-mediated use2 hybrid using next-gen sequencing, gets away from this old way of tracking use2 hybrid interactions on these single spots and enables pooling, because inside of each yeast cell that has an interaction, you'll have this molecule. And it allows you to pool all your cells in one culture and use sequencing to detect the interactions and does not rely on barcodes. So to see if such a method is really possible, I first added the Cree reporter gene to the yeast and induced Cree expression using a known interaction pair. And I saw the pair was able to induce the expression by RT-PCR, and this is in comparison to the yeast DNA binding domain part. And I next tested whether the hybrid plasmid in these recombined coding sequences were detectable by PCR by using the same interacting pair, which induced Cree expression. And as comparison, I also checked for this in cells that could not express Cree. And I saw that only in the cells that could express Cree, I was able to detect that recombined coding sequences. Okay. And then finally, I wanted to test whether if I express proteins in the Cree strains and they are not known to interact, do I detect this PCR product? And I did not. So that means that only cells that express Cree from a protein interaction give this PCR product showing the recombined coding sequences of the interacting proteins. So with that, I went on to do a pilot screen test, and I used this thoroughly screened Arabidopsis gene set. So this is the Arabidopsis interactome subspace, and it consists of 1,500 genes screened against 2,900 genes. And I use this because it was tested in traditional yeast2 hybrid in 6 replicate screens, which amounted to more than 272,000 plates. So this was a labor-intensive, long-term screen. And here, what I did was move the 2,900 genes and the 1,500 genes into the Cry2H-seq expression plasmids. I mated these big pools together in this one culture, selected for cells with interactions, did a plasmid isolation and in bulk, and amplified the recombined coding sequences and sequenced them on a Illumina platform. And I saw that 36,000 protein interactions were in the data. And this was more than 30 times the amount of interactions found in the previous study. And I determined that there were self-activators present. So what are self-activators? These are proteins that acquire mutations in them and can act as their own activation domain so that they can turn this reporter gene system on independent of having this protein interaction. And to control for that, what I did was include this plasmid that has, it's an empty plasmid, so there's no gene in this plasmid. And then in the data, if I ever saw a coding sequence linked to the empty plasmid sequence, I would know it was a self-activator. And I would remove any linked coding sequence fragment from the data if it was detected with that empty plasmid sequence. And after I removed those from the data, I saw 3,500 interactions after the self-activator removal, which is kind of more on par with what's expected. And looking at the overall reproducibility, I saw that Cry2H-seq showed improved overlap between replicate experiments over the traditional yeast2 hybrid, and it was on par with affinity purification mass spec experiments So that's good. It means that the Cry2H-seq replicates are more consistent with each other than previous technology. And I also saw that interactions in more than one screen with greater than or equal to 5 reads in each screen showed 80 to 90% validation in a 1-against-1 used to hybrid retest. So I took the positive interactions I detected and put them in this 1-against-1 test to make sure, to see if how reproducible they are. And it turns out that they are more reproducible than the traditional used to hybrid. Okay, and then in looking at sensitivity, I saw that Cry2H-seq captures more known positive interactions and maintains low detection of random interactions. So here we're looking at sets of positive interactions that were tested in common between the past 2011 assay and Cry2H-seq. And you can see that Cry2H-seq is outperforming the traditional assay in detection of the positive known interactions, and while also maintaining this low detection of a random interaction set. And then I also saw that Cry2H-seq captures more interactions overall than the previous technology. So you can see here, in only 3 replicate experiments, Cry2H-seq was able to detect the 3,500 protein interactions, where the old technology, over 6 replicates, was only able to detect about 1,000 interactions. So in conclusion, the Cry2H-seq technology improves used to hybrid throughput and improves used to hybrid sensitivity. And here we're looking at the amount of time. So this is the old technology. This is the barcode technology in Cry2H-seq. And Cry2H-seq is about 20, it's less time. And it's also less expensive. So this is just comparing the same technologies again and how much money it costs to perform the assay. And in the future, the assay could be optimized by enriching for DNA containing lock sequence, depleting the self-activating yeast cells to reserve more of the sequence space for the real interactions that we care about, and we could also make the assay develop a quantitative measure of interaction strength, which would tell us more about the protein interactions that are happening. But I had one remaining question that I wanted to address, and that was, can Cry2H-seq be applied to address a basic biology question, like the complexity of plant transcription factor networks? Okay, so I'm going to be talking about transcription factors, and that's abbreviated in these slides as TFs, or TF, and I might say TF also. So what are TFs? Transcription factors are proteins that induce or regulate gene expression. So they can induce expression or repress expression. And transcription factors interact, just like our sous chefs do, in different combinations that lead to different expression patterns. So here you can see different combinations of proteins give either like decreased levels of gene expression or increased levels of gene expression, depending on which proteins interact. Okay, and transcription factors plant transcription factors have a lot of complexity. So Arabidopsis has almost 2,500 transcription factors that we know about, belonging to 98 different families, while humans have about 1,600. So you can imagine the different interactions that can come together are a lot greater in plants. And also, transcription factors from large families tend to show similar mutant phenotypes. And these genetic redundancies make genetic experiments challenging. And what that means is if you mutate a transcription factor in one plant, you might see no effect, and then you mutate another one from the same family, and you also see no effect. So in order to see the effect and kind of parse out what pathway these transcription factors are part of, you kind of have to do, all these experiments to figure that out, and it makes it really challenging. Okay, and they're also really important. So we saw earlier that there was these 18 proteins targeted by the pathogen that affected the immunity, and it turns out that 7 of these proteins are actually transcription factors. But we don't know how transcription factor networks are organized. We don't know how many binary interactions exist, and we don't know which families or family members interact. And so I wanted to know, could Cry2H-seq help address these questions? And what I did was take a set of 2,000 transcription factors from the largest collection of transcription factors that exists, which was generated by Jose Pruneda Paz et al. And I moved this set of transcription factors into the Cry2H-seq expression plasmids and made the two libraries just like we saw before. I mated the libraries together, selected for cells that had 2 plasmids in each cell, and then selected for cells that had interactions. Did the plasmid isolation, amplified the Cree recombined coding sequences, and performed an Illumina library prep and sequenced it. And I did this whole process 10 times, which tested about an estimated 1.25 billion protein combinations. And then I created this custom sequence analysis pipeline because one didn't already exist for identifying the protein interactions. And the first step is to map the reads. So let's kind of remember what we're actually putting on the sequencer here. It's these whole pieces of DNA. So these are the PCR products, and there are about 400 to 8,000 base pairs depending on the size of the coding sequences. And they get fragmented to these 300 base pair pieces. And then the sequencer generates reads from each side of these fragments. And so I map the reads to this custom genome, and you can see that most of the reads map to Arabidopsis and the primer sequence, which makes sense based on what we're putting on the sequencer. The pipeline then filters for high-quality mapped reads and pairs the reads and then removes clonal fragments. Oops. And then the pipeline selects for fragments that show the correct DNA, the DNA is in the correct direction. So because these recombination products, because the coding sequences always come together in this way, where one is in a forward direction and the other one is inverted, we can select for fragments that are mapping exactly to that orientation and distinguish those from fragments that are coming from within the gene body. And then we can also use size as a filter. So, okay, because we know the fragments are a certain size. And then the pipeline counts the fragments where one read is mapping to a different gene than the other read, so that we get fragments that look like this. And the fragments are counted, and then a basal fragment cutoff is applied. And to determine the basal fragment cutoff, what I did was sequence the same library twice, once at low coverage and once at high coverage. And what I saw is that fragments that only had 1 or 2 reads at the low coverage were not always reproduced in the high coverage library. So I decided that fragments have to be present in 3 or more counts to be considered an interaction. So after doing all that, from the 10 replicate experiments, 10.9 million fragments mapped to 173,000 different interactions. I computationally removed self-activators, and that gave 1.4 million fragments, which mapped to 8,577 different protein interactions. And I called this the Arabidopsis transcription factor, or Arabidopsis thaliana transcription factor network 1. And this is what it looks like. Everyone can make sense of that, right? So this was just like a quick way to visualize what the data looks like. And here you see all these different colored circles. The different colors represent different transcription factor families. And the circles are made up of tiny dots, which are the transcription factors themselves. And the gray lines are the interactions between the different transcription factors. And since that's really hard to make sense of, what I did was collapse all these interactions on transcription factor families. So instead of having all these little tiny dots making up these circles, I just made that one dot and called it transcription factor family interaction. And then I asked, do transcription factor families show preferential interactions? And to answer that question, I compared ATTFin1 data to data from 10,000 randomly rewired networks. And what that means to generate the randomly rewired networks, what I did was take all these lines and shuffle them all up, and then count the number of family interactions for a random network. And then I got a distribution of family interactions for the random networks, and compared the data from ATTFin1 to this. And if the data that I observed was in the 95th percentile or higher, it was considered significant. And so if it was in the 95th percentile of that, they're colored yellow in this heat map plot. And just for everyone's reference, this is a symmetrical heat map plot. And then if it was in the 99th percentile, it's orange. And if it's highly significant in the 99.9 percentile, it's colored red. And so what you can easily see here is along the diagonal, it's really red. And what that means is that within families, the transcription factors from the same family tend to interact with each other. We also saw these known intra-family dimers. So they're not just like these red ones, but so this NAC family NAC family members are known to interact with each other, just like worky families are known to interact with each other. And then we also saw these known inter-family dimers, so between different families. So the GIF transcription factors are known to interact with the GRF transcription factors. And something interesting that I saw, so looking at unknown or transcription factor interactions that are not previously known, like this big red square here, which is the trihelix ABI3VP1 and the GEBP families. And again, these gray lines represent novel interactions, and the black lines are known interactions. So most of these are novel interactions. And I saw that these family interactions are consistent with cell type specific expression. So in this study done by Villarino et al., they performed RNA-seq experiments on these 3 different cell types in the gynecium. And the gynecium is the egg-developing part of the plant. And what they found in their data is that these 3 cell types showed expression, or so what I saw is that 13 of 14 of these proteins were expressed in all these cell types, but really interesting, in the medial cell type, these 6 proteins show higher abundance in this cell type. So it's possible that there is a complex between these 6 transcription factors that is driving the medial cell type definition. And then another interesting example that I picked out that was interactions where most of them are not known between the G2-like family and the Gras family. And I saw that the G2-like family was mostly these phosphate starvation response-like factors, and the Gras family was mostly these scarecrow-like factors. And interesting, when plants are exposed to low phosphate conditions, the roots start growing these root hairs. And scarecrow is also expressed in the root. And I saw, looking through the literature, that these PHL factors are also expressed in the root, as is this scarecrow-like factor. And so it's potentially, there's complexes forming between these transcription factors that is driving this change in root architecture during low phosphate conditions. And then in this example, this one was really interesting. So this is a known inter-family interaction between the ARF transcription factors and the OXIA transcription factors. So these transcription factors are known to be involved in the auxin response in plants, and auxin is a plant hormone. That's why we see all these different black lines here, because these are known interactions. What is not known, or has not been really found in the plant world yet, is all these gray lines, these new interactions between oxIAA proteins and other transcription factors from different families. For instance, the IAA17 factor seems to be interacting with these TCP transcription factors a lot more frequently than other IAA proteins. And then the IAA11 factor interacts with these two proteins, two transcription factors that are involved in plant defense. And that was interesting because in rice, the IAA10 homolog in a genetic experiment was found to be involved in defense response. Okay, so in conclusion, transcription factors show preferential family interactions. Integrating this cell type specific data with ATTFin1 showed potential complexes driving organ development. In this expanded network view shows redundantly or genetically redundant proteins like the IAA proteins may actually have specific roles. And some future work that could be done is integrating ATTFin1 data with TF DNA binding site data and expression data to uncover these cell-specific regulatory networks. And then also developing Cry2HC for non-model organisms would be a really huge optimization in the assay. This would enable protein interactome maps to be generated for different species, and that would be really powerful. And actually, I had started working on that a couple of years ago, and Renee in the ECR lab is continuing that work. But I kind of had a burning question that I wanted to answer, and that was because the Cry2HC technology wasn't there yet. I wanted to know how existing omics tools could be applied to address a practical present-day challenge. And the present-day challenge I'm going to talk that I've been really interested in is global ocean change. So, because there's a lot of confusion over whether climate change is happening and like what kind of effects it's having. I really wanted to try to understand this and like how things are responding to it, so that I could help maybe educate about it. And to do this, I took advantage of an internship opportunity up at NOAA, is the National Oceanographic and Atmospheric Administration, at their Northwest Fisheries Science Center in Seattle, and that's where I've been for the past year doing this internship. And what I'm showing here is since 1958, that atmospheric carbon dioxide has been increasing, and ocean carbon dioxide has been increasing with it, as well as the ocean pH decreasing. And this is called, the ocean pH decrease is called ocean acidification. And since the 1850s, which was the beginning of the Industrial Revolution, global average ocean surface water has decreased 0.11 pH, which correlates to, or which means an increase, a 30% increase in hydrogen ions. And that's because every year, 25% of human-caused carbon dioxide emissions gets absorbed by the ocean. And that makes more carbonic acid form, and that's what's causing this increase in hydrogen ions and decrease in ocean surface pH. Also, something that's happening with global ocean change is the water is actually becoming warmer. because of the greenhouse gas effect. And warm water does not dissolve oxygen as well. So we see pH and oxygen varying. So pH is decreasing and oxygen is also decreasing over time. Okay. And so, yeah, this problem is affecting a lot of animals that live in the ocean, and that's kind of concerning because we care about certain ones, especially ones that are ecologically and economically important, like the Dungeness crab. The Dungeness crab brings in about $200 million annually and is considered the highest valued fishery on the West Coast. And the Dungeness crab feed on bivalves, and bivalves show sensitivity to low pH water. And because of that, this modeling study done by NOAA has predicted a decline over the next 50 years in biomass and catch for the Dungeness crab. But there's also direct effects that we think might happen to Dungeness crab. So adult crabs have shown physiological and behavioral responses at these temperature, pH, and oxygen levels, and these are kind of extreme conditions that people have tested in the lab so far. And for reference, here is today's current ocean conditions, and this is what's predicted for the year 2100. So that's why it's important that we kind of understand how crabs might respond so that we can prepare our fisheries and everything for the future. And we think that this ocean change is most likely going to affect crabs during this early developmental time period in their lives. Because during this time, the crabs are using a lot of their energy to grow and just become larger animals. So we think that if they're also dealing with stress, that they may not be able to grow as well. Okay, and the Ocean Acidification Lab at the Northwest Fisheries Science Center did a study on crabs that were freshly hatched and were developing into this zoea larval stage, and they saw that the larvae, when they were exposed to this low pH, they did not survive as well, and they also showed delayed development. And so I got to work on this follow-up experiment, which was looking at how does low pH and low oxygen affect early stage juvenile crabs. And here, what they did was collect crabs from Puget Sound, put them into these aquarium jars, and put them onto this mobile ocean acidification treatment system, and then rear them in these different conditions: normal pH, normal oxygen, low pH, and low pH, low oxygen. And they did this experiment over 30 days, and they saw no survival or obvious developmental effects. So how are the crabs coping physiologically? And 15 crabs from each different treatment were sent for metabolomics analysis at the UC Davis Metabolomics Core. And remember, this is the food that is made at the end of the meal, the metabolites. And from this data, we can infer the activity of different biological pathways. Okay, and what I found in looking at this data, so there's 60 different metabolomes, one for each crab, and 15 crabs per treatment. These are heat maps showing the different levels of compounds that were significantly affected by the treatments. So on the right side of the heat map, we see so these are all different metabolites, and these ones are all different lipids, known compounds and unknown compounds. And these are the different treatments across the top. So this is the normal treatment, low oxygen, low pH, and low oxygen, low pH. And if we can read this by, if we look at like this one, this is threonine, we see that it shows low levels in the normal condition, high levels in the low oxygen, low in the low pH, and high-ish in the low pH, low oxygen. But in general, we can see this whole group of compounds increases in low oxygen, while this group of compounds decreases in low oxygen. This group of compounds increases in low pH, et cetera. And so this is really cool that we see these treatment-specific responses, but in order to think about what biology is happening, we have to understand what all these changes in metabolite levels mean. And to do that, I did this biochemical pathway analysis, which is super obvious here, right? You can see all these molecules clustered together, and they end up grouping together based on chemical similarity. So that we have lipids over here, sugars, amino acids, et cetera. And to kind of see what's important here, these green ones now are the metabolites that were in the heat map that were significantly affected. And in general, the amino acid group tended to be the most affected. There was like about 27 different metabolites from this group. And so that's what I'm going to focus on in the next slide. So these are significantly affected amino acids. And here we're looking at the same compounds, only the shapes are now a little different. Blue compounds mean that they showed decreased abundance, and yellow means they showed increased abundance. If they're white, it means they didn't change in the low oxygen, low pH, and then the combined treatment. And what's important to look at here is that energy conservation seems to be happening in the low oxygen treatment group because cysteine degradation was upregulated or was increased because cysteine and cysteine and all these other metabolites were showing decreased abundance. And also because of that, it seems like glutathione synthesis might be decreased because the cysteine is down and the glutamic acid is up. And lysine degradation seems to be decreased. And these three things are actually conserved hypoxia responses in other animals. And in the low pH condition, it seems that energy generation is happening because aspartic acid and malic acid are decreased while citric acid and glutaric acid are increased. So taken together, that kind of suggests that the citric acid cycle might be having increased activity. And that actually makes sense because when animals or cells are challenged with a pH, they need to maintain this gradient in order to keep carbon dioxide coming out. So in order to keep respiring, they need to actually use these proton pumps to keep the integrity of their ion gradients. And these proton pumps or different ion channels require ATP, which is the energy generated by the citric acid cycle. And in the combined condition, we see in general that the low oxygen response tends to dominate. This network looks more similar to the low oxygen network. And we can see synergistic effects on lysine degradation because here lysine is yellow, where in these ones it's not. And we can see synergistic effects on aromatic amino acid metabolism. And I think that has to do with hydroxylases because those tend to be inhibited during stress response in other animals also. So what does all this mean for the crab? We said that low oxygen is a more dominant stressor. Low pH causes less overall physical change or physiological change. And some conserved hypoxia and stress responses are amplified in the low oxygen and low pH conditions. But we still don't know what are the long-term consequences of this. How long can these physiological responses actually be sustained? And will compensation mechanisms lead to things like reduced body size or muscle mass? And then we need to know, would these results even hold true when the crab is also stressed by the increase in ocean temperature or other factors like pollution? So some follow-up work is to perform longer ocean acidification experiments, combine metabolomics data with genomic and expression data, look at genetic associations in natural populations exposed to a spectrum of ocean acidification conditions. So that way we can kind of link what genes might be underlying these physiological responses, and then examine tissue-specific metabolomes like the gill. So this data was looking at whole animal metabolome. If we just look at the gill, maybe we're going to see a stronger signal of the physiology that's happening in that tissue. So some final conclusions that I came to during my PhD is that biology is a result of molecular network activity, and that it's interactions between genes, transcripts, proteins, and chemical compounds. And that omics data sets allow us to see these networks, but they're only as good, the data are only as good as the techniques used to generate them. And there's power in integrating omics data sets from multiple techniques and integrating these, the data from the different biological components can illustrate this bigger biology picture. And that our knowledge and analyses depend on the data that's available. So we need to keep generating data in order to keep understanding the biology. All right, so I hope that I have inspired you to think in the systems level perspective, and I hope you're able to use this perspective in the future to help your understanding of the world that we live in. And with that, I'd like to thank Joe Ecker for having me in his lab for 6.5 years and being a great advisor and like always, sharing these great ideas, and it's been a really great experience. And I'd like to thank Mary Galley, who helped a lot in setting up the Cry2H-seq assay from the very, very beginning and teaching me all about cloning and all the magic. And then Renee Garza, who has been so amazing in helping with the transcription factor screening and just all kinds of support inside and outside of the lab. And then the entire Ecker lab, who's been my lab family for a really long time now. And everyone's it's just been really great. And then I want to thank Salk researchers in the Lundblad lab and some other PIs at Salk, Socket, and Julie, who have helped advise network stuff, and the plant biology department for all their support during our meetings and everything. And then UC researchers, Anne Rexendra Carvunis, who's now at University of Pitt, and Hannah Carter, who's at UCSD, who have helped a lot with the networks analysis ideas, and the Fiend lab at UC Davis, who did our metabolomics processing and software development for that. And then my committee, who have given me lots of great advice during this entire time: Joanne Corey, Jose Pruneda-Pass, Terry Gasterlin, and Trey Eidecker. And the folks at the NOAA Northwest Fisheries Science Center, who have helped a lot, or pretty much guided me through my transition into ecology and applying omics in ecology. And Kristen Nichols helped with the data analysis, and Paul McElhaney with the experiments and the running the moats and all kinds of advice also. And Shalyn Bush for her really great input. She's part of the Ocean Acidification Lab, as well as works in DC. And then Mike Meyer and Danielle Perez actually carried out the crab experiments and monitored the crabs and everything, so that data set wouldn't be possible without them. And they're great guys. And lastly, I'd like to thank my funding, the NSF Graduate Research Fellowship, and the Mary Kay Chapman Foundation. And with that, I will take any questions.
So Oliver Labs is like really one of the best labs for doing metabolite profiling. But how much variation? I didn't get an idea. Years ago we sent samples of different Arabidopsis seeds to Paradigm Genetics, who's now called Metabolomics, and they're like the masters of it. But it was huge amount of variation.
Yes.
I never really sure was it the plant physiology or the assay method that was, So what do you see for the variation?
Yeah, there is a lot of variation in the data. That's totally true. So what I showed here is averages from each treatment. These are average abundance. So if you look at them individually, it can be really hard to pick out the patterns. And to kind of get around the variation, I applied 3 different statistical methods actually to select which metabolites are important. And so I think that's a good way to try to help pick out what's going on.
Consistent through all the consistent part of all the signals that you're getting across all animals.
Right, and using the different statistical methods each one kind of can look at the data a little differently. Like you can use linear regression or a non-parametric method, which is what I did, and it can kind of just pick out different things in the data.
But what about if you put them in like a really well-defined environment? I know it's not nature, but I mean, maybe you want to see them.
Right. So one thing to get around it is getting the animals from the same parent, so that you know where they're coming from, and then raising them in the lab so that you know what they're exposed to try to reduce some of that variation. That's what's happening in the lab right now.
I have a few questions.
Yeah.
Okay. So why do you have so many self-activators?
Why are there so many self-activators? Because the yeast 2 hybrid, let's go back, I'm just going to switch back to that slide really quick. In that assay, you are asking cells to, we're asking cells to grow. We're selecting for cells on this growth media. Because we're asking cells to grow, it's to their benefit to have a mutation to let themselves grow. So you can have get a lot of self-activating proteins because of that. Particularly with the transcription factor screen, there's a high rate of self-activators because a lot of those can have naturally occurring activation domain already.
Did you normal yeast 2 hybrid assay?
In the transcription?
It seemed like you did to me, but I don't really know.
Oh, okay. So I think so like the number, so what happens because when they self-activate, I'm just going to pull up a picture so it's easier to understand. So we're doing this en masse screening. So if you have a self-activating protein, if one of these is a self-activator, it could potentially form 2,900 linkages, right? So then if you have 100 self-activators, that goes up even more. So a lot of the data came from self-activating proteins. Yeah.
Okay. I have one other question.
Yeah.
And so what would happen, do you think, with your interactions if you did them all on the binding site, Did you see different interactions if you really involved the DNA that all these transcription factors are binding to?
Yeah.
Could you do that kind of throughput that involved all the binding sites and everything you put in there too? I don't know.
Maybe. I mean, if you could include an additional plasmid that had different promoter sequence in it and put maybe a different reporter gene. I don't know. I have to think about that. I think it's possible to do some, at least yeast 1 hybrid technology with this. Sometimes having the DNA around does change the way the proteins bind to the DNA. I'm not sure about how it changes their interaction because in the yeast 2 hybrid system, right, they're already anchoring on DNA, so I don't know if that already helps kind of stabilize what they do normally or not, but it would be cool. Anyone, go ahead, I'm sorry.
So what proportion of the protein interactions did you find in only one direction versus finding it in both directions? A interacts with B and B interacts with A.
Yeah, I think so. Most of them were in one direction. Maybe most of them were in one direction, and I think 25% were found in both directions. I think.
So if it were just random, A and B interact, you would expect kind of a 50-50. You would expect everything to be found in both directions if you sequence sufficiently deeply and if you mix sufficiently much. But perhaps this was in part because your DB library and your AD library had different sets of genes in them. So there were actually...
You kind of get a one side, yeah.
So what I'm getting at is it'd be interesting input was in, dB versus AD, how many binary interactions could you have observed, in both directions, and, ask what's the saturation? Have you saturated your
Right.
space, or if you added, if you waited longer, did more, would you build an even bigger network?
Yes. So let me, oh, cool. Okay. So this is like this kind of the saturation estimate over the number of replicates that were done. And then I guess this one's not shown. So I have another figure in the paper that shows like what we estimate to be saturation. And we see that in 10 replicates, it's about 50% saturated. So if we did more replicates, we could potentially detect, more pairs and reach the saturation.
So that was one question. And then, now, my next question is over in the low O2, low pH area. What were the differences in the deltas in pH and in oxygen? And how does that compare to what people find in the ocean being changed versus physiological changes? How were the deltas picked?
Yeah. So those are picked. I'm just going to pull up one. Okay. So these are like today's conditions. The conditions that were picked in the experiment were kind of meant to mimic these 2100 conditions, the future conditions. The pH used in the experiment, I believe, was 7.4, which is down here. And that is something that maybe the crab might experience like very, very briefly now, maybe for like in just such a temporary time, but in the future, we think that it's going to be more in that range. So the experiment, by keeping it at this like constant low pH, is kind of to get an idea of how they're going to do when their environment changes to be like that. And then the oxygen was, I think, about 30%. So I think the oxygen was like lower than what's predicted in the future, but it's possible that because there are these like fluctuations that in the future it could go down to 30% during one of those fluctuations. And we want to know.
So you were testing them really at the lower bound, the lowest lower bound of their what they experience physiologically. So, and you didn't see a difference in development. And then
Over 30
oxygen
days.
was, about a third lower. A third lower? Or 2/3?
Than it is normally. Yeah, probably like a third lower than it is normally, I think.
So in a way, your hypoxia stress was a stronger stress, and you also saw a stronger signal. And an interesting question might be if one repeated the experiment at even lower pH conditions.
Yeah.
You'd be increasing the pH stress. Because if you go forward to your heat map data, what you see is that the oxygen differences tend to persist into the low FOX, the double low, but the pH differences seem to recover. And there are cases where the hypoxia is enhanced by the low pH. You could go through the whole thing.
Yeah.
Especially in your lipids, you have this lipid response down here where you see a little bit in the hypoxic, you see a little bit in the acidic, but when you put them both together, you really see a difference.
Something different happens, yes.
Indicating that there could be a spectrum of hypoxia levels and pH levels that remain to be explored.
Yeah, and I think different things happen, like kind of maybe depending on like the amount of time that the animal's dealing with it, because I think you can decrease some function like glutathione synthesis for a certain amount of time, but eventually you're going to need that, right, to be an antioxidant. So there's going to be some trade-offs that happen over a longer period of time, and it will be really interesting to follow up on some of those. See.
And it would also be interesting to look at the eggs themselves. So
Yes.
taking eggs and looking at how they hatch. So the previous experiment was on the larvae, or was it on
So they did actually look at that. And one other thing I wanted to mention is that there was a lower pH condition in this experiment. They looked at 7.1, and that also had this same effect. I just didn't include it because this is more similar to what I was looking at. And they did look at the hatch rate, I believe, and they didn't see a difference in the hatch rate, of the eggs. The difference that they saw was in the development and survival of the larvae. Sorry, go ahead, Wolfgang. Oh.
Well, go for it.
Well, we're going to have a chance to ask questions afterwards.
Oh, okay.
Okay, good.
Okay.
In that case.
Okay, so going back to yeast 2 hybrid.
Yeah.
So I was wondering, so there are like some very, like quite a large number of experimentally very well studied transcription factors interactions, like the mast box during flower development, or the birds and short-root scarecrow in development.
Yeah.
So have you looked at this? Have you established like a false negative rate of your screen, in your large screen?
Of what we're missing, of the interactions that we're missing.
Very well studied.
Yeah. So let me show. I'm just going to, is that in here? No. I'm just going to talk about a general, the observation, and it was that, like, we were recapitulating about a third of known interactions. So there's still 2/3 that we're not capturing yet. And it can be because our assay is only 50% saturated. It could be because the proteins don't express well in yeast, or the interaction doesn't happen in the yeast.
But
Or
the screen saturation you could like assess, because you know which proteins are in your screen, right?
the what? Sorry.
The saturation of your screen you could assess.
Right, it was 50%. Right, yeah. So the saturation was like 50%. Is that, what do you mean?
So I was just wondering, like from all the potential like things why you could miss them, whether you have checked whether, like, let's say from those matchbox, all these pairs were actually in your screen.
Oh, whether they were in there. Yeah.
Okay, so then like you missed 2/3.
Right, of ones that we could have detected. Yeah. And then another reason is because we're doing this like amplification. So if any of them have problems amplifying, they're not going to be detected. Do you know what I mean? So there's some caveats of the assay, but in general, it can just capture, a bigger amount of data or whatever.